I've been asked to kind of keep it short. This is a case study, so I'm going to give you the general ideas for potential discussion during the panel. It's also a little bit of a shift from previous talks in terms of talking about regulatory and some of the different ways of looking at evidence. So I'll go quickly through this. First, disclosures. Biofortis is a contract research organization. As such, we don't sell products per se. We have received funding from ILSI North America for TFA evidence mapping, which you'll see, I'm just going to touch on that today. And then the American Frozen Foods Association for review of TFA literature, but neither of those had policy positions, et cetera, and no other disclosures related to this topic. The case study is that the FDA in November 2008 published a notice tentatively determining that partially hydrogenated oils are no longer GRAS under any condition of use in food, and therefore are food additives subject to FDA approval. And this was key because it's the first notice of its type, and also PHOs have been in the food supply since, some people say, the 1930s, certainly since the 1950s. They've been well established in the food supply. They're one of the reasons we have a vegetable-based margarines, et cetera. And they were brought into the food supply to replace butter or to provide an alternative to butter and to saturated fat. So this has gotten a lot of attention because it's looking at things in a very different way. So some of the, in terms of destination, some of the questions that this raised is that nutrition research is now being used for risk assessment. And we've seen that in various areas. This is the case example here. In the past, it's been the function of toxicologists with respect to the GRAS process, and they have a risk assessment procedure in place that's science-based. And so we want to look at what does this look like from that perspective as well as from the nutrition science perspective. And it raises the question, how do we determine safety? You know, not discussions about, you know, health preventative diets, potentially preventative for chronic disease, but safety. It's a very different endpoint when you think of it in terms of safety for nutrients versus toxins. We're seeing more examples of lower level exposures as well. I'm going to show you where we are with TFA. It's a low level exposure example. And we have limited data at low-level exposures, and it's really hard to get data at low-level exposures. So how do you deal with those types of situations? Because huge gaps in evidence. And how do we partner with experts, such as toxicologists, in the future, and how can we use what's best in both fields? Okay, so to kind of get our heads around this, and I know Kevin mentioned trans fats. Trans fats are unsaturated fats that have a trans bond instead of a cis bond. The structural difference then is that the trans fat looks a little more like a saturated fat, so it's a physical difference in the molecule. I mentioned that trans fats came into play, industrial trans fats, in terms of margarine as a replacement for butter. There are different trans fats in ruminant versus vegetable-based trans fats, and you'll see that trans fat is a class of fatty acids. It's not just one molecule. And so different sources of oils will have different ratios of different types of trans fats. That's an area that really is I think, just emerging in terms of our understanding of that. And the question is, do different trans fats have different activities? There have been literature indicating that ruminant trans fats may not function the same because it's vaccinic acid is the main trans fat in ruminants. So you'll see that when we go forward on this, we're going to talk more about the industrial-based trans fats. But there's still a class, and it's still a variety of trans fats that may have different activities. The production of trans fats, from an industrial perspective, trans fats are actually generated during oil processing, high temperature oil processing, and found in non-hydrogenated oils. And so many of the oils, when they go through deodorization and bleaching, et cetera, you'll get trans fat formation. What we're talking about are PHOs, which actually come into play when there's an actual hydrogenation process off of this production. And that's also, hydrogenation is also used for saturated fatty acids. And where that comes into play is you also have different sources of trans fats. We have ruminant trans fats, we have non-hydrogenated oils having some level of trans fats, we have saturated fatty acids having some level of trans fats, and then we have partially hydrogenated oils. And the issue has been that PHOs generally have a higher amount of trans fats because that's the purpose of hydrogenation in those oils. They have a higher amount in the actual oil itself, But whether that means a higher amount in the food product is going to depend on how much of that PHO is actually added to the food versus how much of the other components are in the food as well. Because now you've got a non-hydrogenated oil, you've got, if you add a very small amount of PHO, which is actually the current use of PHOs in most foods right now are at very small levels for processing aids, for instance, you might not have the majority of the fat coming from that PHO process. The other aspect to consider here is that you will always have some trans fat in a PHO component that comes from the non-hydrogenated process itself. So trying to understand what's from PHO versus what's from the industrial process of the other trans fats is actually, I'd say, a work in progress. I mean, if you really say, well, what happens when you don't partially hydrogenate and what would happen in the food supply, that's a good question. Okay. So let's talk a little bit about how we do clinical trials and look at evidence. There's two common protocols for evaluating fatty acids. Because fatty acids are caloric components, usually you do an isoenergenic diet or some calorie replacement. So you can replace with carbohydrate. And the other way to replace, which we're going to talk more about, is fatty acid with another fatty acid. So in terms of looking at the trans fat literature, often you're looking at an increase in trans fat as well as a decrease in cis fat in the same group. And now you've got to ask the question, well, does the cis play a positive role and you're really just taking away a positive effect? Or is the trans playing a negative role and the cis is neutral, or are you getting a little bit of both? So these are really tough studies to do, especially when you're starting to focus on just one component. So these are some of the considerations. So I wanted to show you a little bit of evidence that's actually, this is a study that's used a lot because it may be one of the only dose response studies that are out there. And it's one of the only studies that has a couple different levels of a trans fat component at the levels of intake of current dietary intakes. And there was a pointer up here, yes. Okay, so this was from Alison Lichtenstein's group, published in 1999, and it's still one of our flagship studies from that time. It was actually done as a comparison to butter, but most of the data you'll see from now on that uses this does not use this group. But the initial study was comparing to butter, which had 1.5% energy as a ruminant trans fat. And it had much more, sat fat, so that was the comparison. But to note here is the TFA diets. The background diet, which is non-hydrogenated trans fat contribution, is 0.55% energy. The semi-liquid margarine, 0.91% energy, has trans fat, which is on top of whatever you get from the non-hydrogenated as well as the partial hydrogenation. Now this 0.91% energy, I'm going to link to Kevin's talk. Kevin showed EPI data. He showed the Nurses' Health Study data. 0.91% energy was quintile 1. 2% energy was quintile 5, is that right? And the higher one here, soft margarine, is 3.3% energy. So what I'm going to show you in terms of current intakes is that these are the range that we're looking at for current intakes. And so that's how, if you think about the data he showed, that data lies here. But this data is also, was representative of earlier trans fat intakes prior to some of the activities that we've had with labeling. So with respect to the results, this is the results straight out of the paper that compared it to butter, and you can see the soybean oil was much improved in terms of effects on LDL cholesterol, similar to semi-liquid margarine. These were not significantly different from each other, but there was a slight, visual difference here. And then you can see this sort of dose response as you increase up here in particular. Okay, so this identified that, well, there really is an effect of trans fats on cholesterol at higher intake levels. And the IOM did look at different fatty acid intakes, including trans fats, in their 2002-2005 reference intakes. They actually concluded there's a positive linear trend between trans fatty acid intake and LDL cholesterol, and therefore an increased risk of CHD. The upper level was not set because any incremental increase of trans fatty acid increases CHD risk. This statement has been used over and over again, and I want to go through some things really quick, so I'm going to go past this real quick, except that just to point out that they did also indicate that you could not totally avoid trans fats because you'd have to change the diet to such an extent that you would change other nutrient intakes. So and that there are unknown, unquantifiable health risks with those other changes as well. This was the data that was used for linear intakes. People have seen this over and over again. It's from Asherio. It's some of the comments that have come up from this data, though, because this did show a linear intake. This point down here is the Liechtenstein data, which is the 2 lower intake levels. What you need to do in much of this data, because LDL cholesterol levels differ on the control groups, is you then look at a change over a comparator. So that's how the data is looked at. So this is a change. This is LDL to HDL ratio, not just LDL. But down here it says percent of energy from fat. It's actually the change of energy, the change of trans fat in the soft margarine over the change in trans fat from, or over the soft margarine over the non-hydrogenated soybean oil. So this really represents 0.91% energy intake, but it's a comparator of the two. And that's been one of the conversations. The other is that this graph compares an increase in trans fat with a decrease in cis fat. So it's really a cis to trans ratio graph. And I think that's where the majority of the data that's been looked at has been looked at in that way. And again, it's really difficult to try to take fat in isolation like you do another toxin and just add it, because you're adding calories as well. So this is some of the complexity of looking at the data. But I point out this because this is the area of low exposure intake. And you'll see there's one data point on this graph. We have a few more data points now, but not too many, but this is where the data came from. And the idea of this group was, this group said 0% energy change should be a 0% change in LDL and HDL. Therefore, they set the base of this graph at 0, so that the line went through 0 no matter what, and that's what gave them a linear trend. But a lot of discussion has come up, is it really linear in that low region? And that's what we're going to talk about in the last part of the talk. And I've got to move really quickly now. Dietary Guidelines Committee 2005 was advisory committee, was the first and one of the only committees to actually look at the data and say, can we set a level, like an amount that would be a threshold level of some sort? And they came up with less than 1% energy as the level as a guidance, but they also indicated that there really is a very limited data down low. The FDA put in trans fat labeling based on the relationship with cholesterol in 2003 to 2006. Why this is important to this talk, and again I'll move through this pretty quickly, is simply that since 2003 we've seen a big decrease in the current population in terms of intake of trans fat. There's been a lot of reformulations. Majority of that was a replacement with cis oil because it was the liquid oils. Today we have different strategies have to be used for trans fats. Depending on whether they were used for more of a shortening or solid replacement versus a liquid oil replacement. Much of the liquid oil and the cis type replacement has been done. So today we're looking at replacements and changes in technology that would bring in sat fat for the most part in replacing partially hydrogenated oils. But you can see that in the FDA's assessment of trans fat intake, it's in there somewhere, there it is today is the US population is about 0.43% energy from an industrial trans fat. So intakes have come way down. They were up into the 6% or higher in the 1990s. So they've come way down. So this is the level that's relevant to today's intake. And that is, of course, when you look at the Nurses' Health Study and some of the inflammation data, it's down in the quintile 1 range. So we go back to the PHO notice of there's no threshold intake level that was indicated in that notice, and the PHOs are basically unsafe at any level, or TFA, and this is based on the TFA data. So very quickly, ILSI then commissioned a risk assessment process, because when you deal with GRAS and deal with safety, a lot of times the toxicologist then will do a risk assessment process, and I'm going to be speaking about that with this work having been done from another group that is Terra, which I'll talk about their work that we did in partnership. We did the evidence, pulling the evidence together. They actually did the assessment. But you've seen this a little bit already. There's an exposure assessment. I talked a little bit about exposure. But what we're going to talk about is hazard characterization and dose response. Can you actually do a true toxicology assessment? In the last 20 years in the toxicology field, historically, people did this linear regression. You take high levels, you say, well, we don't have data at low level, but we'll do a linear regression. And that has changed in the toxicology field in the last 20 years. And there's been a lot of discussion. When I got into this topic, I looked at the literature and there was just a lot of discussion on why and rationale behind the change. So the current standard is there's a non-threshold considered when you have a cancer endpoint or when you have a mechanism or an understanding that one molecule can make a change in such a way that that would lead to something that's unsafe. If one molecule can make a difference, then it would be a non-threshold and you do a linear regression. But if you have anything else, you look at other literature first. You do a hazard characterization to understand the biology. And from that, you ask the question, is there any evidence that there would be a threshold? Is there any biological evidence in the way this molecule functions that you would need a threshold before you'd see an effect? This is very well discussed in Romberg's Critical Review Toxicology article in 2011. There is still some pushback on the idea that there are such things as thresholds and that from a safety perspective you should always do a linear regression. And Romberg put together a good set of arguments, pretty strong statements saying assuming that every chemical is toxic at high exposures and linear at low level exposures does not comport to modern day scientific knowledge of biology. Romberg went through and kind of identified all of the pros and cons. And so if you really want a good discussion, I think this is a really good one. But the current standard from EPA and National Research Council is to look at the literature first or look at the evidence, which is usually cell culture, animal, and human evidence if you have it. And for trans fat, there's little bits of all of that. For a mode of action. And this is the work that Tara did. Tara is an independent nonprofit science group that does toxicological assessments. And there's actually going to be a poster on this work, so I'll go quickly through the conclusions and you can go to the poster on Tuesday and get all the details. But basically you look at a mode of action, which is not a mechanism of action, as they will point out, but it basically identifies those key events that that it looks like there's evidence of an effect from. And in doing the mode of action work for trans fats, which again is a lot of animal data, cell culture data, and there was human data as well. Not all of it supports, but the preponderance of evidence supports that trans fats do have an effect on VLDL going to LDL. And that's actually one of the key places of the effect of increasing LDL from trans fats at the higher level intakes. It also decreases LDL-IDL clearance through LDL receptor and CETP. So in summary, there's evidence for increasing VLDL production, reducing triglyceride uptake and metabolism, and increasing recycling of LDL-C and CETP. The overall shape of the dose-response curves, though, for these particular key events and these particular activity areas is not linear. And that's where a toxicologist would come in and say, okay, the biological evidence says that if you're influencing something in these places, it would not be linear, and you really need to use certain types of modeling when you look at the data to look at linear and non-linear effects. And there are feedback loops and homeostatic controls as well. So there's no evidence to expect an interaction directly with DNA, which means a linear effect would not be the default for looking at this evidence. So adverse effects would occur only when homeostasis is perturbed. So if we go back to the, and I only have a few more slides, you'll be happy to know. If we go back to the data, this is a publication. It's the most recent publication looking at a linear effect, and this group did only look for a linear regression. This was from FDA scientists. This was not cited by the FDA notice, as it turns out, but it was the most recent evidence. It also is the most recent evidence that also has the most lower intake data points. And you'll notice there's 3 data points. They concluded there was no evidence of a threshold, but did not use the Bayesian modeling that a toxicologist approach would use. And it's really interesting that when they actually did not set the intercept at 0, and they just said, well, we'll do a linear regression on all the data that we can pull together that fit their criteria for data. And again, it was a cis to trans for the most part. This is that relationship, not just trans alone. When they did a linear regression, they actually had an intercept. So they didn't go to 0, they went to an intercept. Which is also interesting because, you know, why would 0% trans fat still have an intercept on a graph? So you really do need to kind of ask that about this. But you can see that there's only 3 data points down there. We were asked by ILSI to do the evidence assessment and pull together any evidence that we could at low intake levels. And one of the differences in the data points, and this is just the low part of the graph that again is on the poster, one of the differences in the evidence that we have is we looked at evidence that wasn't just about a trans-fat relationship, but it was, dietary intakes where trans fat was documented in a percent energy perspective in oil-to-oil comparisons. But they may have been, in some cases, there were things like high oleic oil, high oleic soybean compared to regular soybean. So they just happen to have differences in trans fat in these low intake levels. Similar to Kevin's talk, though, the majority of our low-level intakes were control groups. And so they may have been two different low-level control groups or sat-fat diets. So they were not, one of the caveats is there isn't a lot of evidence out there in low-level intakes that really is true dose response for trans fats. So you're kind of pulling what you can about dietary intakes at low levels. So that's a caveat. But you can see that the data is pretty scattered around zero. The closest fit linear or nonlinear model, flexible model is, you see that this is the top of the 95% confidence interval is the top line. The other line that starts about 1.5% energy is the actual best fit line. What this evidence would say, and of course there's more of the graph on the other side, but I wanted to show you this aspect of it, is that the best fit says that there would be a threshold at about a change in TFA, and again we're looking at a change over baselines of 0 to 0.7% in energy intake and comparator, at about 1.5% energy intake. So there is evidence from this type of approach for a threshold effect of some level, and that threshold is around the areas of intake of current intakes right now. So in summary, and I think I'm almost on time, applying the risk assessment techniques to a macronutrient can help in understanding from a new perspective. And I think one of the calls to action here is that nutrition scientists and toxicologists, we really need to be talking to each other, especially about risk assessment, because we're now using both types of data and both approaches and looking at risk assessments and health effects. And so we have a lot to offer from both aspects. But the substantial data gaps that exist, look at this graph. I mean, we looked everywhere we could to find data of low-level intakes. And you can see there's a huge gap in data right where you need to have data if you really want to know if there's a threshold or not. So there's like a whole percent energy from 0.5 to 1.5, which is really around our current intake levels. We have a total gap of evidence. And this is change over background, but still, it's important place for us to be looking, and we just don't have a lot of evidence. The other aspects of this is that most of the evidence we have was not designed to ask a question about safety. It was designed to ask questions at high levels, and these happen to be the groups that had the lower level intake that represented what we are currently consuming. We found in the evidence assessment that there was a lack of description of processing methods as well. And I think Tricia talked about looking at food sources and some of these other aspects in future evidence. We found a lack of, there'd be studies that use margarine, but they didn't say if they truly partially hydrogenate or not. Now you assume they do. You assume at high levels of TFA that's what you have to do. But at these low-level intakes, you'd like to know if this is about a process that we're talking about, we'd like to know that the evidence came from data that was developed on ingredients having undergone that process. And a lot of that information is simply not there. Also, there were many different intake units. There are other articles that have intakes at gram levels, but didn't put it into context of percent energy. And we look from a regulatory and policy at percent energy. So those are some of the comments for future studies. We really need an updated assessment, though, of TFA intake, one that takes into account what's coming truly from a PHO process versus non-hydrogenated background of TFA as well as ruminant TFA. Because we just don't have good updated sources. The FDA's done a nice job of the recent data that I showed you, but there's a lack of information on, again, processing. How much comes from processing, and what would our intake look like if these ingredients actually went away and they were substituted by, say, a sat fat or something? How much TFA would you actually have in your background diet? That's a key question that I don't really have an answer to. So those would be some of the needs, and I just want to say, here's the team that worked on the projects. Melissa Vincent will be at the poster later to answer a lot of questions about the toxicology side. And I want to thank everybody for listening.